देखिए पीसीआर देखिए ना, पीसीआर मतलब क्या है, पॉलीमरेज चेन रिएक्शन है ना। पीसीआर मीन्स पॉलीमरेज चेन रिएक्शन, पॉलीमरेज चेन रिएक्शन, चेन रिएक्शन। ठीक, फर्स्ट वी हैव टू नो व्हाई वी परफॉर्म द पीसीआर। ओके, व्हाई वी परफॉर्म द पीसीआर? सपोज you collect the gene of interest from a particular murderer site, okay, or you want to match this gene along with the suspected person, okay. So this gene is very small in quantity, no, this gene is very small in quantity. You isolate the gene from the particular cell or few number of cells. So you do not want to lose the identity of your target gene or which you have isolated from the murder site. You never want to it become damaged. So that first you have to produce the multiple copy. You have to produce multiple copies. ठीक. Then you perform the gel electrophoresis. When you get the multiple copies of this DNA or polymerase, when you get the multiple copies of the DNA, that is the variable number of tandem repeat. I think day before yesterday I told variable number of tandem repeat means in our genome there have so many repeating sequence. ठीक, this is known as the variable number of tandem repeats or known as the VNTR. Okay, and this variable number of tandem repeat become differentiate from person to person. Okay, so variable number of tandem repeat you have isolated from the particular cell which obtained from the murderer site, and this variable number of tandem repeat we have to match the suspected person. Then you have to think, suppose there are the 10 suspected person, among the 10 suspected person who is the murderer, उसके साथ ए जो है उसका ही डीएनए है, उसके साथ मैच हो जाएगा. कहाँ मैच होगा? जेल इलेक्ट्रोफोरेसिस में मैच होगा. ठीक, मतलब सपोज यू गिव हियर द suspected person DNA, suspected person 1 DNA, then you give here the suspected person 2 DNA, give here the suspected person 3 DNA, and here you give the murderers from murderer site with DNA you have isolate, you give the VNTRs of this DNA. ठीक, सब में मतलब सस्पेक्टेड पर्सन 1 का भी VNTR दिए हो, मतलब फ्रेगमेंट्स दिए हो, 2 का भी फ्रेगमेंट्स दिए हो, 3 का भी फ्रेगमेंट दिए हो. Then you just switch on the gel. Okay, when you switch on the gel, this is known as the anode. Okay, ठीक. DNA is the negatively charged because it contains the phosphate. So negatively charged DNA will move towards the positively charged anode. Anode is known as the positive electrode. Okay, then you have to look, this is the from murderer site you get. तो murderer site का भी DNA ऐसा मूवमेंट करेंगे. Small size DNA will move faster and large size DNA will move slower. Okay, suppose suspected 2 is the murderer, तो उसके साथ ये VNTR मैच हो जाएगा. ठीक, और इसका सोचो सस्पेक्टेड पर्सन 3 का यहीं आया, यहीं आया, यहीं आया, लेकिन यहीं पर देख रहे हो मैच हो गया.
तो जो सस्पेक्टेड पर्सन है वो अपना जो अलग हो जाएगा, सबसे अलग रहेगा.
मतलब सस्पेक्टेड पर्सन 3 और 4 वो क्या रहेंगे? अलग हो जाएंगे. लेकिन सस्पेक्टेड पर्सन 2 ही तो वही जाके मर्डर किए थे ना.
जो मर्डर रहेगा वो सबसे अलग रहेगा.
जो मर्डरर रहेगा तो मर्डरर साइड से जो डीएनए मिला वो इतना सस्पेक्टेड पर्सन वो साथ में चलेगा, क्योंकि ना दोनों डीएनए तो सेम है भाई. अभी समझ में आया? दोनों डीएनए तो सेम है. ठीक, तो we never any kinds of work in any kinds of work we never lost the identity of the particular gene. We have to produce the multiple copy of this gene, then we perform the gel electrophoresis. Okay. Or second thing, that is the one target of the PCR. Second thing, second target is suppose you getting the gene of interest from the pancreas cell. Okay. You getting the insulin gene. Okay. So how many cell you cut from the particular human body? Few numbers of cell. Few number of cell you get from the human body during the surgery. Okay. Then you never want. Suppose you have to produce the multiple number of clone. Okay. Suppose you look.
बोल रहे हैं ना मैं किसी को खराब हो गई थी मैं तो वही के साथ आएंगे
This is the gene of interest. So you getting the gene of interest and you getting the gene of interest from the pancreas cell, and you want to produce multiple number of clone. You want to add this gene of interest along with the so many vector. First you have to produce the multiple copy, then you can able to add with the vector, na? Okay. So for that you have to produce the multiple copy of this gene of interest outside of the cell, means in a particular machine. This machine is known as the PCR machine. Okay, understood? That is the main target. ठीक? So main target or significance of the PCR is to get the multiple copy of our target gene or any DNA. We get the multiple copy of our target gene or DNA outside of the cell is known as the in vitro condition. If I write the definition, bahar. We want to replicate our gene or DNA outside of our cell, means in vitro condition, in vitro condition. Okay. Then we going to the processes of PCR. Okay. If we discuss the process of PCR, you look, this is our DNA. All of you know DNA contain the two strand, and the two strand are the anti-parallel in nature. This is the 3' to 5', and this one is the 5' to 3', and in between there have the hydrogen bond. That is your the template DNA.
First one is the
Template DNA means from which DNA you want to produce the multiple
strand.
copy. Okay, template strand from this DNA, or this is the main DNA from which DNA you want to produce the multiple copy. Okay, that is the older DNA. Okay. Then first what we have to do, we have to apply the huge temperature. We have to apply the high temperature to break the hydrogen bond between the two strands. First we have to break, so that two strands will be separate. Okay, this stage is known as the denaturation. Okay, in denaturation stage, two strands become separate from each other.
92 to 95 degrees Celsius.
And which temperature? At which temperature, beta?
92 to 95
92 to 95 degrees centigrade. 92 degrees centigrade to 95 degrees centigrade temperature. When you apply the high temperature, then hydrogen bond will be break up. And when hydrogen bond will be removed, two strands become separate from each other. 5' to 3'. Next, we know when we read the DNA replication, we read that first this is the strand, this is the 3' to 5', this is the 5' to 3', this is the replication form. So at the starting of the DNA replication, we learned that first RNA primase primate enzyme come and produce short stretches of RNA. This is known as the short stretches of RNA. Then main enzyme is the DNA polymerase 3. Then main enzyme add the new nucleotide from this new strand. Okay. So first work is to produce the small primer or small RNA. ठीक. Then DNA polymerase add the new nucleotide and produce the new strand. This is the function by the DNA pol 3 or DNA polymerase 3. First function, this is the primer, and this primer is produced by the primase enzyme. Primer is produced by the primase enzyme. ठीक. Same thing we have to apply here because replication is going on here, na? Okay. So first we have to add the primer. Okay. So first we have to add the primer. This is the primer. Always primer have to be produced by 5 prime. This is the strand. This template strand is 3 prime to 5 prime, and new DNA should be synthesized from 5 prime to 3 prime. समझ में आ रहा है ना? New DNA is synthesized from 5 prime to 3 prime. Okay. Next you look. Next, this is the new DNA strand. Then new DNA strand will be produced. First produce the primer. Okay. This stage is known as the annealing. Okay. This stage first denaturation, second is known as the annealing. This is denaturation. Actually इसमें नहीं करना चाहिए था. सपोज ये डिनेचर होने के बाद ये स्टैंड बना, उसके बाद एनिलिंग हुआ. ये एनिलिंग और एक स्टेप करना चाहिए था, एनिलिंग. सो इन एनिलिंग स्टेप 2 primer are joined along with the template strand. Then in annealing we use the less temperature, like that 40 to 45 degree centigrade temperature. Okay. Just I look the temperature. Then elongation. Okay. This is the separated strand, and this is the primer, and this is the 3 prime to 5 prime. This is the 5 prime to 3 prime. And when during the elongation process you have to add the DNA polymerase. But you have to add which DNA polymerase? You have to add the DNA polymerase which can tolerate the high temperature. Okay. And which DNA polymerase able to tolerate the high temperature? Which DNA polymerase have to isolate from the bacteria who are live in the high temperature? Which bacteria? Thermus aquaticus bacteria. From Thermus aquaticus bacteria we isolate Taq polymerase. Taq polymerase. ठीक. We isolate the Taq polymerase from the Thermus aquaticus bacteria. Then this Thermus Taq polymerase, it is a DNA polymerase. This Taq polymerase actually add the new nucleotide and produce the new strand. Add the new nucleotide and produce the new strand. Okay. And this is the 5 prime, this is the 3 prime, and in between there have the hydrogen bond. Hydrogen bond is formed in between 2 strands. Okay. Now you look, from one strand we produce the 2 strands. Okay. Even half an hour में हम बहुत बिलियंस कॉपी बना सकते हैं. मशीन में डाले हैं हाफ एन आवर, विथ इन हाफ एन आवर. ठीक. तो देखिए फास्ट क्या है, पूरा टेक्निक को अगर देखें, इफ वी वांट टू आइसोलेट द जीन ऑफ इंटरेस्ट फ्रॉम द प्लांट सेल, ओके, then plant cell is made up of cellulose, because cell wall of the plant cell is made up of cellulose. Cellulose. No? This one is the cellulose. Yes. Plant cell wall is made up of cellulose. Okay. So we add the cellulase enzyme. Cellulase enzyme to break the cellulose, because cell wall of the plants is made up of the cellulose. Okay. If you use the bacteria, if you want to get the gene of interest from the bacteria, you use the chitinase. Chitinase.
Chitinase, which helps to break down the chitin.
Chitin, because cell wall of the bacteria is made up of chitin. And next,
Animal cell.
animal cell membrane is made up of the
Protein and lipase.
protein and lipid. Protein and lipid. So to break the protein, use the proteinase. To
And
break the
for
lipids,
lipid,
lipase. Then you get the complete genome, because when you use those enzymes, then cell membrane, cell wall will be break down, then nuclear membrane also will be break down, then you get the gene of interest. Then you have to separate this gene of interest, because ये gene of interest, sorry gene of interest बोल रहा हूँ, ये जो genome complete genome सेल का कबाड़ा मतलब मिक्सचर के साथ मिला हुआ है, इसमें सेल है और ऊपर में डीएनए है, इसके साथ मिला हुआ है. तो टू आइसोलेट द डीएनए फ्रॉम द मिक्सचर ऑफ सेल, और सारे सेलुलर ऑर्गेनेली तो है, you have to add the ethanol, chilled ethanol. Chilled ethanol help in precipitation of
DNA.
Okay, and when DNA become precipitate, it become float on the test tube or float on the petri dish. Basically test tube we use करते हैं, ठीक. When we DNA become float, then slowly we isolate the DNA. This process is known as the elution. Okay, so what is elution? By applying the chilled ethanol, by applying the chilled
DNA from the
sample, precipitating DNA from the mixture of the cellular organelle. This precipitation of the DNA or separation of DNA, this process of separation is known as the elution. Then when you get the complete genome, then you add the DNAs. यहीं पर क्वेश्चन आता है, डीएनएज को पहले यूज करेंगे ना सेलुलेज को पहले यूज करेंगे? तो सेल वॉल को ब्रेक डाउन होगा तब ना डीएनएज यूज करोगे, ठीक. ये क्वेश्चन आता है. फर्स्ट वी ब्रेक डाउन द सेल मेम्ब्रेन, सेल वॉल जो भी है, देन वी गेट द कंप्लीट जीनोम, देन बाय द प्रोसेस ऑफ इल्यूशन वी गेट द कंप्लीट जीनोम, देन वी ऐड द डीएनएज टू काट द डीएनए to get the gene of interest. Gene of DNH is the restriction endonuclease which cut the DNA inside the DNA. Okay, then you get the gene of interest, from the complete genome. Then you have to cut the vector with same restriction enzyme and same restriction enzyme. Then you have to join this gene of interest along with the vector by the DNA ligase.
Molecular glue.
That is the molecular glue. Here you use the molecular glue.
DNA ligase.
Molecular glue, DNA ligase. Then you add this cDNA. Or you insert this cDNA inside the host cell. Host may be plant cell, host may be bacteria cell, or host may be animals. ठीक. तो बायोरिएक्टर मतलब क्या है? इट इज अ लार्ज वेसल. Quickly look, it is a large vessel. Inside this large vessel you insert the raw material. Raw material means what is the food of the bacteria. Okay, you insert the raw material, you insert the sufficient oxygen, you apply the you insert the oxygen, you apply the proper acid, proper base, proper pH, and you have to recombinant DNA transferment. Which bacteria have the recombinant DNA? This bacteria you insert in this bioreactor to get your desirable product. Okay, bioreactor means it is a large vessel. In this vessel we insert the bacteria and we insert our raw material, and this raw material is consumed by the bacteria and give the product. Two type of bioreactor, one is known as the radiated tank, another one is the sparse stereated tank. Stereated tank बायोरिएक्टर का बेस देखिए, दोनों का डिफरेंस देखिए क्विकली. इसका बेस होगा कार्प, इसका बेस होगा फ्लैट. प्लेन। इसमें क्या होगा, ऊपर में मोटर होगा, मोटर के साथ लगा हुआ रहेगा फोम बेकर। देयर हैव द फोम बेकर, क्योंकि ना जितना घूमेगा उतना फोम होगा, फोम को ब्रेक करना है। देन फ्लैट बेस इम्पेलर, this flat base impeller and culture growth, it help to mix the oxygen along with the solution. Okay, then here you look acid and base for the pH control, and from the bottom you give the air, because if you not give the oxygen then bacteria will not grow. Okay, and difference here you look, यही एयर को ऊपर से दिया जा रहा है। यहीं पर स्पार्च टैंक में देखो क्या है, स्टेरिएटेड एयर बबल आर स्पार्च टू इंक्रीज द सरफेस एरिया ऑफ ऑक्सीजन ट्रांसफर। ऊपर से दिया जा रहा है, यही नीचे से। ठीक, ये डिफरेंस है। इसका बेस कार्प है, इसका बेस प्लेन। एंड देन एंड आफ्टर सम डेज यू गेट द प्रोडक्ट फ्रॉम द बायोरिएक्टर। Okay,
for the
downstreaming process we separate our product,
by favorable
product. Okay, downstreaming process means it is the isolation and purification of the product, because in the bioreactor there have the bacteria also, nutrient solution also, that means raw material also, and another waste material also. So by which process you isolate your desirable protein or desirable product from another substance is known as the downstreaming process. Downstreaming process, there are the three parts: separation of the desirable product, सारे मटेरियल से सेपरेट कर लिए हो, प्यूरिफिकेशन ऑफ द प्रोडक्ट, उसके साथ और कुछ मिला हुआ रहेगा तो रिएक्शन कर देगा हमारा बॉडी में, यू हैव टू प्यूरिफाई, देन फॉर्मुलेशन विथ प्रिजर्वेटिव, भाई प्रिजर्वेटिव के साथ देंगे, then it will long lasting until and unless it will not be used. ठीक, then you just packed it and sell in the market. Okay, that is known as the downstreaming process. तो बायोरिएक्टर और डाउनस्ट्रीमिंग प्रोसेस दोनों एक साथ जुड़ा हुआ है। Inside the bioreactor we produce the product, and in the downstreaming process we isolate the product and purify the product, separate the product,
separate the product,
purify the product, add the preservative, okay, and
preservatives,
packed it in a bottle and sell it,
into the market.
sell in the market. Okay, this is the complete.
Yes.